Now it's my pleasure to introduce Artem CHERKASOV, Professor of Medicine at the University of British Columbia in Canada and Director of Therapeutics Development at Vancouver Prostate Center. Computer-aided drug discovery, artificial intelligence, chemoinformatics, precision cancer therapies, and this guy is a serial grant writer. Sometimes he gets money, so he got a rather small amount of money, $80 million. You know, that's not enough to achieve the transfer of even a second-class footballer, not to mention Cristiano Ronaldo. So therefore, he had no choice but to do useful things with this money, and he licensed 8 drug candidates to big pharma or spin-off companies. So he got some awards like the Achievement Award for Excellence in Fundamental Science, Tier 1 Canada Research Chair in Precision Cancer Drug Discovery, and of course, the highest consecration of all of them, he is a constant participant at the CS3 School in Chemoinformatics. So here you go.
Thank you so much, Dragas. So first of all, of course, I'd like to thank Sasha for having me here. Times and times, it's one of my favorite places to be. And so happy to be here and happy to present. So a lot of talks on these days, we're kind of traveling back in time and summarizing the field, which again shows me that the field is mature enough that we can start telling stories. And so today I'm going to tell you a few stories in the context of AI, and will be 3 parts. When I'll talk about past, how we've been doing AI all days, and then how it's done now, and where are we going. So we're going to go from computer-aided drug discovery to human-aided drug discovery very soon, when basically I firmly believe AI will be really doing it while experts will be guiding the process. So talking about past, in old days, when we talk about AI or use of AI, really we talked about neural networks. And we started using neural networks in early 2000 to deal with antimicrobial peptides. Idea was that these peptides are extremely good antibiotics, anywhere 7 to 20 amino acids long, and people have been using bioinformatics to deal with them. So they would shuffle these letters in every possible way, try to find patterns, try to substitute, and they had decent antibiotics, but they really hit the wall. And at some point I said, why don't we look at the atomic level resolution? 9 amino acids long, it's still a small molecule. So we actually built them and basically expanded them into conventional QSR descriptors, passed them through pretty simple shallow neural network, and labeled with activity. Very simple at the time approach, but it worked. And it really, really changed the way antimicrobial peptide discovery actually is ever since. So we changed the paradigm because this was working just wonderfully, and we published a paper saying that AI was used to discover new drugs, which was, again, state of the art at the time, while, again, there is not much AI about neural networks, we all know that. Nonetheless, when I presented this at ACS in Boston, it caused so much waves that I was interviewed by Boston Globe, and I said that in 2007, AI will be developing drugs, and they published a piece in the morning press. So next thing I know, I'm in a TMZ magazine. Because at the time when I said that AI will be developing drugs in 2007, it was the same league news as Britney Spears not wearing underwear and abusing her children. And so this is probably the highlight of my scientific career. We were on the front page of TMZ. So that's how I got famous first time. So obviously, after such an achievement, I couldn't stay in the infectious disease field, really, and I moved to cancer. And so I moved specifically in the prostate cancer and started working on the biggest problem there still is drug resistance. So here's a very standard scenario for prostate cancer. So cancer onsets and tumor grows up because it's an animal model. So you see it goes up 3,500%. And then you start treatment. At the time, the drug to go was biglutamide from AstraZeneca. So you administer that and it goes down. So really, drug really works. But in 20% cases, roughly at 121st day, it was plus minus a couple of days all the time, cancer comes back. And it comes back at what we call castration resistant or CRPC, prostate cancer form, uncurable. and enzalutamide, recrutamide wouldn't work. So question was what's happening here at day 121. And so it was established that what happened was one single mutation, W741C. And this is a very unique scenario. As far as I know, no other targets behave like this as androgen receptor. So that single mutant turned drug into agonist. So more drug you give, more cancer you get. And so it became obvious that we had to change the way we treat patients, we watch them, we administer drugs. And enzalutamide was a blockbuster drug, so total sales were close to $14 billion. And yet that mutation, again, at 20%, actually killed patients. So very quickly, a couple of other drugs emerged from Pfizer and Janssen, where they started selling close to $3 billion a year. And these drugs, enzalutamide and apalutamide, didn't activate that mutant. So basically they worked. Problem was, for us chemists, we can tell it's the same drug. So this is really copycats, which big pharma do, with the warhead which binds to the target is the same. And then I said, well, they may deal with that particular mutation, but I cannot believe that these particular two drugs will not inherit the same problem. It's just not going to happen. And so we decided to start watching for mutations. And we adopted a technology which was called cell-free DNA and next-gen sequencing, next-generation sequencing. So basically, the idea is that tumors are messy, and tumor shreds a lot of genetic material in the bloodstream. And next-generation sequencing is such a sensitive technology, you can pick up a single copy of mutated DNA out of 1 milliliter of blood. Again, 1 copy per 1 milliliter is enough for that technology. So what we started doing, we would take blood sample for patients before they go into treatment, do the analysis, how an androgen receptor looks like, how AR is mutated or non-mutated. Typically it's not. And then we start treatment, and then we keep watching what's changing in androgen receptor. And once we started it, we found close to 30 mutants, not just that one I mentioned, but 28, 29 others. And they all been focused on exon 8 here as the genomic organization of androgen receptor, because that's the ligand binding domain. That's where the action, the chemistry happens. That's where steroids bind. And those mutants, we see they're all over the exon 8, starting from position 700 all the way 900 plus. So all over the map. But again, they did a pretty basic thing and mapped them all on 3D. And next thing you know, all those mutants are actually around steroid binding site. Then I got even more suspicious. I said, you know what, probably they're all doing something. And again, being chemist, working with biologists, a lot of time I ask stupid questions and suggest stupid things which they never do. And the results sometimes pretty spectacular. So what I said, why don't we make all these mutants physically? Make them, put them in a lab and just screen them against these drugs. Pretty simple idea. And we did. And the story started unfolding. So we started watching what's going on in the patient, started watching what's coming up in the AR mutations. So here's an actual story, 56 years old man with pretty severe prostate cancer. So PSA is major prostate cancer marker. So it goes up to 16, it's like fully blown cancer, but there is no mutation in biopsy and AR, everything is fine. And about day 250, that gentleman was really treated with biclutamide, with Cassidex, that AstraZeneca drug. Was doing very well, pretty much he was cured. At day 200, things got worse and he really developed CRPC. And metastasis went all the way to the spine, to the bones and everything. So things looked very, very bad. But we checked his mutations and there was not that W741C, it wasn't there. So I said, let's start watching for everything else, those 30 mutants. Next thing we know, that gentleman had 2 mutations previously unreported. 889 and 896. And when we made those mutants in a lab and put them into that drug biclutamide, we see the drug actually activates. So the more drug that guy was getting, the worse he was turning to. And I said, if nothing else, we have to stop. Better, we have to switch the drug. And so, again, it opens a lot of questions, but main thing was, we switched that guy to Enza, and things really got back to normal. And it went so well that even metastasis disappeared. So really, we went back to close to PSA 0. Metastasis, you can see, disappeared from the bones, and the guy actually got another decent year of life. Unfortunately, of course, cancer plays a lot of nasty games. He got worse again. And then he really went back into fully blown CRPC and we couldn't do anything about it. But what's more even really striking about it, when we look at his blood again, like what was it, after 200 days. So here at day 200, he had these two pink dominant mutants, if you've seen the previous slide. After enzalutamide was administered, he got really well. And then again, things got back to worse. He developed 4 new mutants. So the pink ones we call, they shrank down to 20%. So 80% of new mutations previously unseen in that guy, now they're enza resistant. So same scenario, more drug you give, more cancer you get. Now, second drug here. Again, opens a lot of different opportunities and questions and dark stories. Basically, this is classically tells you that medicine should be personalized. So every patient on a treatment should be watched what's happening. Mutations should be really studied and functionalized. You see what's happening to the drug, because cancer is a very smart beast. And here you can see like fighting it in real time. And when we started looking at all those 30 mutants, we made them all and started looking. The picture was even darker. So reality is every single of those mutants was doing something. So here at some point, even if drug works with some known mutation, at some point you look down, boom, new mutations come up, drug stops working again. To make things work worse, I asked my people to look into steroids. And what we found that some mutations which don't really turn the drugs into agonists, yet do other things, such as estrogen, testosterone, progesterone, and cortisol start activating androgen receptor. Totally non-specific, non-conventional steroids start working. Some mutation made androgen receptor sensitive to testosterone, like 150%. So every single mutant was doing something. That was the bottom line. But of course, as much as it looks dark and difficult, but obviously this is the holy grail for machine learning too. You see how much data it is here. So I asked one of my students to build AI, already at the time, deep model. And so what we've done, and was pretty early work too, 2 years ago, which is in this field, is infinite time. So we mixed in one neural network descriptors from protein and descriptors from small molecule. And we fed them together in a hot vector, which wasn't like normalized binary vector. And we've been training network for whether it's activated or deactivated or mixed or non-reacting response. And we used that previous massive set of data to basically train this neural net, and it was working pretty okay. So your standard ROC of 90%, few times cross-validated, so everything looked pretty decent, which opens a lot of opportunities again. So now we can use this AI trained on patient samples to watch what's happening in patients. So when new mutation emerges, we can say, predict whether it's what it's gonna do with prostate cancer. So things look pretty manageable already, but clearly, another take-home message still is that this part of a protein is so prone to mutations. So cancer gets so smart about it that we actually probably have to change the way we target that protein. So as I said, all those drugs, they all act in this exon 8. That's where the mutations happen. That's where all those marketed drugs are binding. That's where steroids sit. So this is where the chemistry happens. But the actual active site of that protein is exons 2 and 3. That's where DNA binding domain is. And that's in fact what the active site of transcription factors is. So when I remember presenting this to doctors, and I said, like, you guys been looking into basically allosteric regulation of this thing for 60 years, because this is the active site. That's where you should be hitting. And just the audience went silent for a couple of minutes. And that's what we've done. So, and this is old story. This is something I'm not going to present. This last time, 4 years ago, that's what I was talking about. But so just to mention that instead of going after exon 8, we went after DNA binding domain. And we've developed drugs here. And again, this story is already in the history. So we just look at the DNA binding domain of this receptor. And this is the dimer sitting on DNA. This is the active site. And we did the usual thing. So we took it apart. We built surfaces. We analyzed the binding site, drugability, and we did docking. Again, the usual stuff. It's just nobody ever tried that part of the protein. We did. And we at the end of the docking, we found this molecule, and we went through just 5 million compounds at the time. So we docked 5 million, we found this compound, and we licensed this to Roche for $142 million. So that was and still is the biggest Canadian academia deal. So everything worked fine in 2015, and no AI was really needed. So that was the status quo, and we've been pretty successful using this pipeline. So we would start with 5 to 10 million molecule sync, then we would do docking, then we would run some consensus scoring, then we would pass it to the lab, they'll do assay, and everything was great. So we're getting grants, we're getting drug leads, everyone is happy, we get tons of papers, status quo was pretty comfortable. And then, well, there is a slide which most of the speakers in these last few days been presenting in different shape, that something really happened around 2015. So the number of compounds in zinc went from, what was it, 5-10 million, all of a sudden 1.4 billion. And even though we had pretty decent computational facilities, for us to pass this 5 million would be a couple of weeks. Now all of a sudden, to pass the entire zinc, it would be 2.5 years, even though we had probably 800 CPUs. So all of a sudden, the status quo is disrupted, and we're no longer really controlling the situation. Not to mention that, again, these numbers keep coming up, that possible number of compounds can be as high as Google. Well, more realistically, 10 to the power of 23, which is synthetically feasible, in the power of 60, but still, even 1.4 wasn't doable. And in real space is already 40 billion when you enumerate stereoisomers. So 40 billion, 1 billion, and 10 million, a pretty big gaps in there. So then I remember we went back to a paper I published in 2006. So in 2006, I published a paper in JMed Chem called Progressive Docking, when I said, why we keep docking? Why don't we start predict docking scores? Because you can dock just small fraction of a database and then use, again, the simple ANN, you break 2D representation of a molecule into bits, you feed it in ANN, and you predict docking score. You don't have to do 3D, you don't have to do enumeration, you don't have to do rotation, you don't have to do docking, nothing, just predict. But nobody cared back in 2006, so paper was completely ignored. And the reason is because at the time, zinc was probably 500,000 molecules, and every group would have some decent CPU cluster, so docking zinc wasn't a problem back in 2006. But now it is. And so we decided to revisit the progressive docking, and we really made few modifications. So we threw in obviously more hidden layers, because now we can. We went through more modern descriptors. So originally we worked on Max. Now obviously we went into 2-3 radii Morgan, which is more universal. And also we tried different algorithms. But more importantly, the main thing we changed, the progressive docking, was rejecting bad molecules. So you predict in front of docking, which don't look good, you remove them, and that's it. This time, since it's augmented drug discovery, deep models, they like positive data. So we started augmenting the training based with predicted actives. Really, in reality, how it looks, you go through cycles. And you do the first step iteration. You dock 1% of a database. You predict the remaining ones just based on the neural network. And then you take all the predicted actives, put it back into training, retrain again, and on and on and on and on. So on average, we go 11 to 13 iterations. And as you can see, the number of molecules in the database dropped from 1.4 billion, that was that zinc, right? Down to 300 million, step one. In principle, that's where we can stop. Problem is then you have to dock remaining 300 million still. But then we just keep slashing and keep iterating, and 11 iterations, we usually end up with 20, 30 million molecules, which are dockable. But those 30 million out of 1.4 billion are pretty enriched compounds. So it's basically, they've been predicting to be active 11 times. So all of a sudden, things look pretty decent. And again, as often happens in life, something else happened. And that something else was COVID. So, but before I go there, we built this new pipeline. So, but now, rather than docking 10 million molecules and screening this whole thing, we're basically putting GNN on top of the process. So there is a GNN module which sits on top of the workflow and keeps learning. And it keeps learning what's docked, what's not, and it's iterative process, and docking became 100 times faster. And we checked this enrichment for hits on pretty standard 12 family of drug targets, starting with our favorite AR all the way to all other things. And so we could see that the enrichment, depending of course how thin you cut the top, up to 4,000 fold. So 4,000 is a pretty good enrichment for hits. And so we started playing with this approach, with this new paradigm. And then something happened, which was COVID. So COVID came to life, and very quickly a lot of potential targets emerged. So up to 25 different proteins been crystallized. And we picked it up as docking base. So we took an MPRO and we decided to run deep docking on MPRO. That was our first real target. So we take MPRO, 3CL protease from SARS-CoV-2 virus, and then we pass the entire sink. So that was the biggest docking ever reported, because the biggest before was from Shoykit group when they docked to 130 million and published in 2019 paper in Nature. And UCSF has a pretty decent cluster there. So, but we passed 1.4 billion using our deep docking, and we found hits. And more importantly, when we look at the SARS-CoV-1 main protease as a benchmark, again, which wasn't, now we know it's not good benchmarking, but at the time we didn't have anything else, we could see that that's where the random scores are, if you just do glide. That's where known inhibitors are. You see they're actually not very good at all. And that's where deep docking landed. So you see that we typically shifted everything 1 to 2 units to the negative area. So deep docking seemed to be really, really enriching. So we ran 3CL Pro and we identified 1,000 hits. And it was February of 2020. Actually, nobody knew about this virus yet. And so then I decided we have to publish this. We have to give the society these 1,000 molecules to try. But we never publish papers without experimental validation. We never do. So I called them in and I said, Sasha, there is this new virus you might be interested in. And we actually already ran our deep docking. We ran 1.4 billion. And we have 1,000 molecules we'd like to publish in your journal, but it has to be fast. And Sasha goes, virus we want, yes, sure, go for it. And it took us a month, of course, we went through the usual review, but I'm sure Sasha twisted some arms because we published paper without experimental validation. And we published it on the day when pandemics was declared, March 11. So March 11, we already had a paper in a real journal, real paper with 1,000 drugs suggested how to treat this thing in the day it happened. And well, I'm sure you're pleased now, this is the most cited paper in Wiley right now. So hopefully we're both happy. But more importantly, and we found the hits. And so these 1,000 compounds, we tested 600 of them, found 30 actives. More pleasantly, that a lot of third-party groups also did the same. So we had people reporting some of these 1,000 compounds being evaluated independently and working and all that. So pretty nice story. But more importantly now, we first ever in the history of chemoinformatics reported $1 billion campaign, billion molecule campaign. I'm thinking dollars all the time. So basically the race for billion docking was on. So Oak Ridge Center, the biggest computing cluster in the world, they threw in 45,000 GPUs, and of course they docked it in one day. Then European supercluster, they used Autodock, and with 27,000 GPUs, they did it in one day. This is as close as to us as possible. This is one of the California universities when they did 8,000 cores, they did it also in 3-4 weeks. This is regular docking mine. We did it in 5 weeks with 60 cores. It was a pilot study. So that's the comparison. That's the difference between docking and deep docking. Of course, we're losing 10% of hits. I should be upfront about it. At the cost of 4,000 fold enrichment, 10% lost, especially in docking, which is completely wasteful exercise, is fine. And this is something which really was bugging me, how wasteful docking is. Because you push the button and you wait for 2 months till it's finished. And then after 2 months, you have a look. Like, come on. Basically, for 2 months, there is a data being generated, some useful stuff to be learned, and you're not seeing it. So basically, deep docking changed that. So this is a progressively self-learning system which capitalizes on everything which happens as it goes on in the real world. So we thought, okay, we can also get access to a certain number of CPUs. How about we dock more than 1 billion? Because these guys are already in the same league with us. So that's where we decided to do a little bit of a future. So we called up Inamin, before I go there, and we decided to dock 40 billion. But before going into that direction, we decided to do something else. So it was already full-blown pandemics, and everyone was really searching for drugs. And I said, we have to not just dock it again, we have to build automated system. We have to build something which will be finding drugs on its own without us. Because next time there is another pandemic, we have to have it in place. So next time there is a COVID, we push the button and we get things done. And the reason I was sort of confident we have to, and we can't do that, because back in 2005, we wrote the Genome Canada grant. This is actual snapshot, it was awarded. So in 2005, we said we have to be ready for emergence of new pandemics. And in fact, we named SARS among other threats which may reemerge. And we said we have to build a knowledge base for the scientific community to treat new infectious agents. And we have to have this in system. Again, fortunately, we're actually funded. And we did build the system, and that's how it looked like. And in the core of that, again, this is another trip in the past, was this picture. That's how we built this grant. For most of you probably not knowing what it is, it's probably 2005 Nature. This is internet. So this is how the first time ever internet was mapped. A group from Notre Dame, led by Aldo Barabasi, they decided, let's look how the internet looks like, because nobody regulated it. It's self-built system. And they wrote the program, the code, and they put it into World Wide Web, and the code mapped the connections. And what emerged out of this was pretty interesting picture. So internet appeared to be super unequal. So you have those hubs highlighted, like few of them, which handle 90% of internet traffic. 99% of other hubs are not well connected. And that turned to be the blueprint of life, scale-free network. And so back in 2005, again, our idea was, let's build this system when we look how proteins interact in bacterial virus. Let's see if it's a scale-free, and if it is, we'll hit in the hub. That was the idea. So we got the grant, as I said, and we conducted 13,000 mass spec experiments, and thus we mapped the protein interaction of Staphylococcus aureus. So here you see there is this core. So there are proteins which are really the business end of the bug, while the most of them are actually not interesting. And when we map the existing drug targets, which is in red, they're like all over the place, but not really in the center. So we decided to use, again, you can call AI or machine learning. Back in 2007, that's how the computers look like. That's how our idea of CAT looked like. But the very idea was right. Again, we would look at the interacting proteins, find the center of this ball, find the target, pass it through docking and whatnot, and some machine learning models, and would find a drug which would be hitting it in the heart. And that's what we've done. So we've developed a drug which was picomolar for MRSA, methicillin-resistant Staph aureus. It really works. So that's how this grant was a success. That's how we put this group together back in 2005. That's how we kind of proved that we can build this system. And so when COVID came to life, we kind of already knew each other. We've done it before. And the protein interaction network's been published very, very quickly from UCSF. So when they map all proteins interacting between virus and the human, within the virus, and so there is a pool of potential drug targets was available. But as I said, we picked up MPRO, and much like all other people did the same in the next few months. And then we decided we're going to run 40 billion and we're going to run it 5 times. So the reason we're running it 5 times, because it was very much us who published this paper called Best Practices of CAT, where we said you have to do the consensus. Because programs are still not perfect. So we all know that docking is best case 10% active. But when 5 docking programs agree on something, probably it's going to be okay. So that's why we decided to do 40 billion in 5 different ways. So we used 5 programs, Glide and ICM and Fred and Autodoc and Fred, I think, is something else. And we ran 40 billion in the mineral space plus zinc, 5 times using 5 programs against 3CL Pro. And then we ended up with 200 billion endpoints. There were no even tools to handle it. So we had to write in the midst of pandemics, tools to even navigate through that. And so we started writing and clustering and coding. An idea was, my desire was that out of 5 different programs, we can build a lot of consensus protocols. So here it is, 28 consensus protocols we tried. So basically after docking 40 billion, with 5 programs, we ended up with 20 million good-looking molecules. Here are 5 different programs from glide to autodock, and probability is blue is more probable, red is less probable, but still this is a cream of the cream. And we started clustering. We see how different programs agree, how different RMSDs overlap. We started throwing in pharmacophores. So basically, with some consensus, protocols would be like 5-6 consecutive steps. So we start congesting for blue. And in some scenarios, we congest down to 40 molecules, from 40 billion to 40. Of course, it's blue. But these are 28 different scenarios, 26 really, and then we also threw in human expert ones. When we did things old way, so there's a guy in a dark room, sitting at the monitor and picking up what he likes. And when we compared these 28 protocols, and these are selected hits, you see only at the very top there is some agreement. So we're using same starting base, 5 docking programs, we just massage them differently. Nonetheless, all over the map. And the last 2, again, that's the human expert. So in order to see what we cooked, we ordered something like a couple thousand compounds, and we placed them through our standard, again, MPRO assay. Not going to go into biology, but typically we start with relatively high throughput enzymatic assay, something which worked, what we put in live virus. So we have live SARS-CoV-2 virus in university. We have level 3 security facilities there. And what worked there, we put even human lung organoids. So we went from hits to reasonably active compounds. And as I said, we ordered 2,500. So these are 28 strategies. And we ordered more or less equally from every one of them. You see, we tried to keep it close to 100. And there are human expert 2 last rows here. So we ordered 2,500. Vendors were able to sell us 1,400. Those got tested. We ended up with 160 plus hits. And as you can see, every strategy delivered something. That's good news. Well, as probably you're not going to be surprised, the human expert did the best. Still, you cannot rule it out. But these automated protocols are encouraging. This is completely automated now. So if you dock hard enough, long enough, with many enough programs, and you squeeze the results in a stringent way with consensus protocols, you end up with hits which pretty much human free. So you can next time build this pipeline, and basically starting with protein interaction network, you can push the button and end up with number of reasonable hits. So we started building this core. That's what we're trying to build at university. When you start with the genomics core, you find how the genes look like. Then you look at the protein interaction network, how things interact within the virus, between virus and the human. Then, of course, structural biology to get the structure we target, our favorite cat with AI guidance of everything. And then assaying, PK/PD, animals, clinical trials. So, but this time we're going to make it really automated. And again, deep docking enables that to become human free. And that's where we kind of going to. And it's not like I mean to scare people, but there are much worse viruses out there than COVID and SARS-CoV-2. There are viruses with 70% lethality, up to 50% lethality. So basically, if these things get transmitted more efficiently, we really need that system. So that's basically the vision, how with rigorous AI, with rigorous consensus docking, we can build relatively automated systems for drug discovery. And that's where we're going to. And with that, of course, I have to acknowledge a lot of people who helped us with companies and granting agencies, because what we proposed was received very, very well. And now we play with the big boys and keep publishing and keep enjoying the use of AI. So with that, I have a few minutes for your questions. Thank you.
Okay, thank you very much. Questions?
So, wonderful work. I think congratulations. So I have a couple of questions. So when we're doing this deep docking, how do you address like the binding side? Sometimes you know it's the side, but the other side, how do you handle that?
This is another beauty I didn't mention about deep docking, which comes back from my old PhD work when I was working on Hammett equation. So basically, when everything else is the same in the system, you focus only on the changing part of the system. So the protein is the same in your docking. You can rule it out. There is no protein. It's ligand based. That's the beauty. So it's really fast and cheap. But in principle, you can, put in protein ligand descriptors, FODE and whatever, and train it better. That will give you probably 5% accuracy, but you will lose all the speed. So there is no protein in this. There is none there.
Thank you for your talk. I have a question. What was the overlap between the expert selected drugs and your protocols? Was the AI able to find things that the human could not see?
Absolutely. Yeah. So if you look at this slide, these are the overlaps. So the expert is the last two columns here. So you see there is some overlap, especially on the top, but there are a lot of other things which came out of non-human.
And do you think it's possible that in the future you could also apply eventually explainable AI to be able to understand and rationally explain those?
We can do it even now, in principle, because there are several talks, like Pavel gave one, how you can actually look under the hood and see what's what. It's possible. We can, again, this can be analyzed for many, many months after that. We've been kind of result oriented. We had to have a drug for COVID. Now we can step back and reflect and look at things for sure.
Thank you for your very inspiring talk. I have a question regarding the applicability domain of the first machine learning model that you trained, because you sample, I don't know, 50,000 or 100,000 compounds, but certainly the applicability domain of the model trained on these molecules doesn't reflect the full data set.
Okay, so that was one of the biggest first questions, how to sample things. Because in principle, we talked to computer science professor if we can cluster it before we can do it. And in fact, that's how I did progressive docking. So I would cluster, since it was small enough, I would cluster it and sample from each cluster. This time it was impossible, and Prof told us there is no way. So we started doing random sampling. And of course, we did smaller benchmarks, smaller sets. And we compared if random goes really bad compared to a cluster. It's the same. So as it turned out, so we just randomly sample, so there is no AD problem here per se, because the rules are simple and clear from the beginning. We sample things as it is, and we get the results as they are. But in general, for instance, when it came to peptide, everyone has own AD application. So I just look plus minus 15% beyond the range of descriptors. That's my AD. And the reason for that, because all experimental numbers are plus minus 15%, something we all should remember. So I just impose the same criteria. So if your descriptor changes from -1 to +1, you can add 115 and plus 115. That's it.
Okay. Thank you very much.
Thank you.
Thank you for your topic.